Hi, everyone. Welcome to this new session of MRM Highlights. My name is Emily McKinnon, and in this episode, we'll be talking to Sebastian Papazoglou and Siawoosh Mohammadi from the University Medical Center Hamburg in Germany. Their paper on biophysically motivated efficient estimation of the spatially isotropic R2 star compartment from a single gradient recalled echo measurement was an editor pick for November. Let's first have a look at their project.
Hi. I'm Sebastian Papazoglou, and it's my pleasure to present one of our projects to you that has been selected as a highlight in magnetic resonance in medicine. This project actually started when Siawoosh Mohammadi was a postdoc in London at the Wellcome Center for Human Neuroimaging together with Nikolaus Weisskopf. However, at that time, they couldn't finish the project, and in the meantime, Siawoosh moved to Hamburg and started his own research group. So when I started as a postdoc in Hamburg in 2017, I inherited this project, and together we finally managed to shape it into a publication in MRM. Due to this, and also due to the scope of the project, there have been many valuable contributions during the course of time from a large number of people, reflected by the long list of co-authors. The project was motivated by the well-known orientation dependence of gradient recalled echo-based R2 star and white matter of the human brain. Here, the classical mono-exponential signal expression leads to an R2 star that is dependent on the local orientation of white matter fibers relative to the main magnetic field of the MR scanner, as color-encoded in the DTI image. In the temporal decay of the signal, this would show up as illustrated schematically by the different slopes of the red and blue curves. The effect could be explained by Wharton or Botel using a biophysical model in which the white matter fibers are represented as hollow cylinders featuring a myelin sheath with transverse anisotropic magnetic susceptibility. Depending on their local orientation, the cylinders cause different microscopic magnetic field distortions, leading to spin dephasing, which impacts R2 star. The least distortions are observed for cylinders parallel to the main field. We use this condition to define R2 star iso, the isotropic part of R2 star. For perpendicular fibers, distortions are strongest, and correspondingly R2 star reaches a maximum. The difference between the red and blue line corresponds to the anisotropic part of R2 star. The problem is that due to this effect, the R2 star value that we observe at a specific location in a sample becomes dependent on the positioning of the sample inside the magnetic field. This in turn limits its utility as a quantitative biomarker. Therefore, a separation of the orientation-dependent part from the orientation-independent part of R2 star would be very helpful. This separation has been achieved by others before us. However, our method differs from these methods in that only a single gradient recalled echo experiment at an arbitrary, unknown orientation of the sample is required. In order to understand our proposed method, we have to look at the explicit signal expressions of the classical model and the HCFM. As shown before, the classical model is linear, with the first order coefficient being the fully orientation-dependent R2 star. In contrast, given that we are in the static dephasing regime and the employed echo times are not too long, a second-order Taylor expansion of the HCFM signal shows that the linear term features no orientation dependence. It could thus be interpreted as the isotropic part of R2 star. In this approximation, the orientation dependence occurs first at second order in time. In order to validate this hypothesis derived from the HCFM, we measured a formalin-fixed ex vivo sample of a human optic chiasm at different orientations relative to the main magnetic field using a custom 2-channel coil at 7T. At each sample orientation, the signal decay was mapped using gradient recalled echo at 16 distinct echo times. Note that our longest echo time exceeded 36 milliseconds, which is the time limit for the static dephasing regime as estimated by Wharton and Botel for in vivo samples. We then applied the classical model and the proposed second-order model to the observed temporal decay. We then analyzed R2 star in two regions of interest inside the left and right optic tracts of the chiasm. Since there we expect the fibers to be organized into bundles with well-defined direction along the optic tracts. We then plotted the mean value inside the region of interest as a function of orientation of the optic tracts in the main magnetic field. For the classical model, we recovered the well-known sine to the power of 4 behavior of R2 star. In contrast, when using the second-order model derived from the HCFM, we found no observable orientation dependence in the first order, which confirmed our hypothesis that at first order we will observe R2 star iso. The second order showed again the known orientation dependence. We further verified our observations by statistical analysis and correlation of the model parameters, which further confirmed our hypothesis. To summarize, we introduced an efficient method to fit R2-star ISO from a single gradient recalled echo experiment, and we verified that our method can in fact remove the orientation dependence of R2-star under the experimental conditions in a fixed human optic chiasm. Future work will have to investigate how the method translates to the in vivo case and to lower field strengths. In particular, local iron could also lead to a higher order time dependence in the temporal decay. Further work is also required to extend the method to less coherent fiber bundles than in the highly aligned tracts of the optic chiasm. Thank you for your attention.
So we're really excited to have you here. Congratulations on your paper. And so to get started, maybe you can tell me a little bit about your path into MRI.
So first of all, great talking to you, Emily. I'm Sebastian, and thank you to MRM for choosing us as highlight. And so my path to MRI is actually there's a little anecdote. So this guy there on the right in my screen, Siawoosh, and we actually wrote our master thesis on condensed matter theory in the same office at the same time in Hamburg. And after finishing our thesis, Siawoosh was a little bit more determined than I think, and he chose to move into MRI, and he warmly suggested considering this to me. And I had a look at these things, and I thought, wow, that's looking cool. And then it happened that I got a PhD position in Berlin. So then the combination of MRI and Berlin convinced me to do this in the first place when I started with MRI 15 years ago.
Wow, time flies, huh?
I have another short thing to say, that when I did this analysis on this for the paper, that's almost now at least 1.5 years ago, and I didn't have the chance to have a deep look again into this stuff from my code and so on. So it might be that I'm not remembering all the details. I hope then that Siawoosh can jump in sometimes at least, but I will do my very, very best.
What about you, Siawoosh?
For me, it was in a time when you still needed to do in Germany either a military service or a civil service. So I was kind of looking, so I just was finishing my master degree in Germany. It was called Diplom at that time. And then I was just Googling and looking for something where I could do civil service instead of military service. And I found this university, and I typed also in physics and electrodynamics and things like that. And then there was a civil service position, basically where my later PhD supervisor kind of was offering a civil service position in diffusion tensor imaging. That was the time before even FSL existed and all the tools. So we were they were using MATLAB and SPM and trying to build up their own DTI fitting tools and so on. That was how I basically got into that topic. And it was kind of cool because using the same kind of mass I was knowing from condensed matter physics, but now applying it to biology. And that was then also I stood there, did my PhD there, and so on.
Oh, that's interesting that we consider that a civic duty, right? So, and then I guess a little bit more specific about this project itself. So can you tell me how this project got started? Because it's been going on for a little while, if I understand that correctly, and how Sebastian, when you picked it up. And
so I can tell you something about when I picked it up. Exactly it was somewhere in 2017, late 2017, beginning 2018, I think. And there was just the opportunity of doing the postdoc together with Siawoosh, and there were a couple of projects and that were available, let's say. And this was one of the projects that I found most interesting. There was another one that I also found very interesting, which came later then, but we started with this one. And Yeah, as I said also on the audio slides, there are many people involved in this project because Siawoosh started in London already with Nicolas Weiskopf, but for this part, I think Siawoosh can tell you definitely more than I can.
So it was basically, I think, shortly after the PNAS paper and the neuroimage paper was published by Wharton and Botel, when Nick was still, I think he was writing his grant, actually. He was writing his ERC grant at that time and was discussing with us these models. And it was really one of these models which was generative. That's what I liked about it. So that's, I think, really the PNAS paper where you start from this hollow cylinder model and describes the signal, that kind of really struck me. And it was first the PNAS paper which I wrote and tried to understand, which took a while. And then I was thinking of, okay, cool. But what is missing totally is the inverse model. So it was just a forward model. So you couldn't really extract from the idea the biophysical model parameters. And that's what I kind of was already thinking of at that time. And then the second paper came, which kind of was already the inverse model, but it wasn't there. So it was, everything was there in the paper, what you needed to do the inverse fit, but it was doing it differently than what I had in mind. So it was kind of a very good starting point to do the project.
I guess talking about the model, can you tell me a little bit on how dependent your results are on the accuracy of this hollow cylinder fiber model? Like, I guess, would things change if this is not an accurate approximation?
I mean, our inverse model is based on the hollow cylinder fiber model. So of course it will depend on how well the hollow cylinder model describes the physical reality.
Mm-hmm.
So if there would be changes, then I would expect also that the inverse model wouldn't work as well as it does in our publication in the project. Yeah.
Yeah. So, for example, would it matter if you have like different T2 relaxation times in the different compartments?
I mean, the signal in the hollow cylinder fiber model consists of three exponential terms. The sum of three exponential terms for the myelin, for the axonal space, and for the extracellular space.
Mm-hmm.
They discard the myelin space. And they just keep the axonal and the extracellular, and they assume that the relaxation time for both is the same.
Mm-hmm.
Right? if you assume that but I think if you assume that there is a difference between the two, then you could still do a very similar derivation of the Taylor expansion, so that the main difference will be somehow that you will have something like the difference of the two relaxation rates occurring somewhere in the expansion. But that I didn't do the calculations, so it's just intuition.
Yeah. Siawoosh, do you want to add anything to that, or?
Yeah, I think as well. So we would need to do the calculation, but I think it's a really interesting question to. That's also maybe the real the cool thing about it is that you have this link to the diffusion and to the community. To, for example, your work there. But I think the bigger point is actually the static dephasing regime here, which kind of in the same model, independent of whether you have different TEs. T2 relaxation, T2 rates in the two compartments or not. You will the one compartment, the extracellular one, will at some point be violated. Violated is the word. Sorry. Will be violated according to the theory, and then basically that's a game changer, because then you get a linear term. Mm-hmm. in the solution, which is orientation dependent.
Okay.
And then, according to the theory, you can't do the extraction of the isotropic part from the linear term and the anisotropic apart from the higher order term anymore, because now the linear term becomes as well orientation dependent.
Okay.
So my gut feeling is that this, or that's basically what we are looking at into more detail at the moment, to kind of find a way to determine the static dephasing regimes in different scenarios, because probably it's different in vivo than ex vivo. Now
that you have these two metrics that are one that's orientation independent and one that's orientation dependent, and obviously the orientation independent one is very interesting as a more of a parameter that's, you know, describing like microstructure or something. Do you think there's any interesting information in the other parameter that is orientation dependent?
Well, and definitely I think so, because when you look into the hollow cylinder fiber model, then you have in this second order term, which is just governing this sine to the power of 4 term, you have lots of microscopic information, and it's like the fiber volume fraction, and I don't remember exactly all the terms, but I remember that there are many of them, like the susceptibilities and so on. And so I think definitely it is a kind of a quantity that is important. So also, I mean, the myelin sheath, if it's healthy, let's say, then it is supposed to have an anisotropic anisotropy. So
correct.
It is also a measure for the integrity of the fibers. Also, I think could be kind of, that's at least my impression.
So do you think, which one do you think would be more interesting then?
I think both would be, but there would be would have some complementary information, right?
Yes.
So.
Oh yeah, for sure.
That's what I mean. But maybe Siawoosh can also say more, and he has always the ideas.
I think this is really, that's the nice thing about the model, that you separate these two. And in my eyes, they are kind of, even though you have an effective one order, so first effective, how you call it? Effective R2 star. But you can kind of disentangle these two parameters, which probably come from different compartments. And that's, I think, the nice thing about it. So you're using one contrast and kind of get two compartments out of it. And I think there was a hint in our data already that they have different features in terms of they are sensitive to different microscopic features, and that was when we looked at the fixation. We had two data sets. One was with, and they were differently fixed, and one was basically PFA 4%, and the other was 3% PFA and 1% glutaldehyde, I think. And this kind of changed the isotropic relaxation rate, but didn't change much the anisotropic part, interestingly.
Oh, interesting.
But that was just one shot, so we had just these two, and I think we reported only one of them. I'm not sure whether we reported the second one at all. But this is something, I think it's just a hint. And it's kind of, it's coming from the other side. Typically, so for example, if you go back to the intraaxonal and extraaxonal T2 relaxation rates, this is the diffusion side, right? Which the diffusion community, or contrast, if you want to call it like this. And here we come from the relaxation rate and also kind of find one which is associated with the fiber, which is orientation dependent, and the other one which is kind of maybe extracellular, I don't know.
You might not be able to answer this question, but have you tried it on like a whole brain, and does the contrast change within white matter, like let's say even just the one that's isotropic?
I think here comes the crux of the model at the moment. So that's what we are at the moment trying to understand. And the goal was to kind of bring an answer for the ISMRM with Frank, but we didn't get there. And I think it's really difficult because, first of all, okay, in vivo, if you want to have it really I think that's the end of the question, right? In vivo 3T whole brain. So there comes, of course, physiology, so that thing moves. You can't go to these high TEs to get really disentangle the parameters. And then there is another mechanism, which is probably totally different in vivo than ex vivo, which is iron. And iron can also have a second order decay term, which then I don't know how that will influence the model actually. So whether it's in vivo actually still valid or not, I don't know. So with the data we looked at until now, we couldn't really, we are not there that we can answer whether it works or not works. So it's much too noisy. So one of the things we looked at for the ISMM now is really the noise, how does it amplify? Because the problem you have is you introduce another parameter into your model. And then if you look at the matrix, which you have to invert kind of, if you just do it as a linear model, and then you can calculate the condition number of that problem, right? And a condition number of, with two parameters, where one would be the offset and the second just the linear decay, which is the classical model, is of course much better than if you have now the third column, which is coming with a TE square, because TE square is not orthogonal to TE. So they are collinear. So the condition number becomes much worse, and then that means that you become much more sensitive to noise. So the noise enhancement, that's what we basically observed for the ISMM abstract, is kind of 5 times higher if you do the second order model than if you do the first order model, and that's where we stand at the moment. So if we just, so first step would be whole brain, fixed tissue. And there, the noise amplification is so high that we are not really sure how good we are. So, but we will work on it. We have also a brain which we rotated and so on, but it'll take a while.
No, yeah, very interesting. Sorry if I kind of made you like spill the beans there on your ISMRM work. So I guess going back to then the paper a little bit. So, so Sebastian, what was the biggest hurdle you had to overcome in this project?
Oh my goodness, there was so much work. I remember that there was so much work to do, and so much working with the code, and also finding just placing the region of interest in I had to see how I should do this, not to prove that I have no control of my experiment, but to find reasonable locations for them. And there was a lot of playing around with these parameters, let's say, and this took a lot of time. And actually, in the end, I think this kind of really turned out to be pretty nice and straightforward. So I had more, let's say, tedious projects in my life than this one. This was pretty nice and pretty compact as well. So this is maybe also the reason why it was why we wrote a note first in MRM, because it's very focused and compact, and yeah. So that's,
yeah, it was an easy, good read.
It was but it was not easy work.
No, that's not. I can imagine.
I don't remember all the things, but there was a lot of.
Were you involved in the preparation of the optic chiasm?
No. That was mainly, I think it was Tobias who did that. Tobias did the measurements, actually. I was more on the analysis side and the modeling side and these kinds of things. And I think it was Tobias, right? Yeah.
I think the preparation was even done by so a few of the other co-authors which are there. Carsten, what is his name, last name? Oh, that's now... He will kill me when he hears that I don't know his last name. Okay, maybe it comes later. Jäger, yeah.
Jäger.
Oh,
exactly.
And Markus Morawski, these were the guys who are doing the histology.
Great.
But can I dive in, because I think there are a few other things here.
Of course.
To my mind. So first of all, the orientation, extracting the orientation. You remember, right? So first of all, you need to realign this, which is already a pain, and then kind of finding the right transformation and just get your head around it. And with all the other problems, don't make a mistake there, because otherwise you kind of lose the whole orientation.
Yeah, of course.
We have to register the optic chiasm onto one first, let's say, theta 0 angle case, and then in order to extract the angles that we have. And this was kind of tricky as well too. Yes. Yeah.
And the second point is region of interest. That reminded me of also another really important limitation of the model. It's of course only looking at parallel cylinders. That's why region of interest. So as soon as you have not a really isotropic part, kind of the model doesn't work anymore. And that's also something for the ISMM, the next one. So that's something we managed to look at a bit.
Oh, great! Lots of teasers here. Sebastian, what do you think the biggest, I guess, takeaway and is from the paper, and what does this bring to the community?
Wow, that's a big question. So for me, I mean, first it was we just for the first time showed one possibility of separating these two terms in with just a single measurement, which was just one thing that is, I think, pretty cool. Of course, we have to show, one has to see how it translates into in vivo and so on, all this ongoing work. And I love this personally love this physical, biophysical approach from a modeling point of view. And yeah, that's one things that I like the most. It's a very, very nice approach that opens the windows to some physical parameters in these measurements.
Siawoosh?
So I think that the community, biophysical modeling community, is kind of trying to get to unify the different contrast mechanisms we have. So that's kind of the holy grail, right? We want to go to you as well, probably with your work with Jensen, where you look coming from the diffusion side and try to estimate the two different relaxation rates, intracellular and extracellular. That's, I think, something similar from the other side. So we are coming from the relaxation side, and we are trying to separate what is probably on a different, I don't know whether it's a different scale. At least it's a different feature, so it's an isotropic part than from the anisotropic part. And I think the anisotropic part here is really the thing which can unify the contrast, because it's orientation dependent. So if you kind of imagine that you find a way to fit gradient recall echo data and diffusion data simultaneously in a unified model, then this here can help you really, because you now have the anisotropic part orientation dependent, which can be estimated from diffusion data, whereas the other parameters, microscopic features, can be estimated maybe from the gradient recall data. So I think this is really what might really be a valuable contribution to the community. If it's a correct model, that's now the question, whether it is still valid in vivo or not, of course.